Good morning everyone. We'll continue with the topic cell cycle and cell division. In the previous class, we have seen the process of mitosis and what are the stages in the mitosis, what are the events that occur in the prophase, metaphase, anaphase and the telophase, along with the significance of the mitosis. So those processes are also being followed, or the same pattern of events also occur in the process of meiosis also. Yes, same processes, but there are certain changes occurring in the events of the cell. So here, yesterday I said that mitosis occurs in the body cells or the somatic cells, where two daughter cells are being formed, which are similar to each other genetically and they are similar to the parent cell also. So they receive the equal number of chromosomes. But while meiosis is known as reductional division, with the first chapter, that is with the first class, I had said you all, where the mother cell divides to form 4 daughter cells, and each daughter cells are having half the number of chromosomes. They are not identical with each other. So we'll continue with the process of meiosis. Meiosis, as earlier said, it is a reductional division where the daughter cells are haploid. They receive only the half number of chromosomes. Those cells which undergo the process of meiosis are known as meiocytes. The cells which undergo the process of meiosis are called as meiocytes. That is, those cells which are involved in the process of meiosis are called as meiocytes. And those cells are reproductive cells. We call them as reproductive cells or germ cells. We call them as reproductive cells or germ cells, where it was been discovered by Strasburger in the plant cell and Fleming in the animal cell, and the term meiosis was coined by Fleming. So in meiosis you will find 2 divisions, that is first division and the second division. That is, in first meiotic division, the same process, that is karyokinesis 1 followed by prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis. At the end of cytokinesis, 2 daughter cells are being formed. So this is about the first process of division, and you have to remember that though it has 2 divisions, only the replication of DNA is only once. Here the nucleus divide twice, but the replication of the DNA is only once. So the nucleus shows 2 divisions as meiosis 1 and meiosis 2. Where at the end of cytokinesis 1, 2 daughter cells are being formed. Those daughter cells again undergo the next second division, that is meiosis 2 followed by karyokinesis 2, prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis. At the last you will get 4 daughter cells. So here some terms are there, so that before going to the process, I like to say you some of the terms that are being used in this topic, that is haploid and diploid. Yes, so in meiosis there are the nucleus divide twice and there are 2 divisions, meiosis 1 and meiosis 2. All the events are similar to as that of mitosis, but these occur only in the reproductive the germ cells. But in this, the prophase 1 has 5 sub-stages and is of longer duration. So we will study in this the prophase 1 with longer duration. So here the parent cell, that is the meiocytes that undergo the process of meiosis, it enters into the process of karyokinesis, then followed by prophase. So in prophase, what are the events that occur or which are the sub-stages of the prophase 1? So there are 2 nuclear divisions, or meiosis is divided into meiosis 1 and meiosis 2. So you can look into the chart given in that where it has mentioned the process. So here, before going to the process, we'll be familiar with the where it occurs. This process occurs in the reproductive cells or the germ cells. If you take in plants, the reproductive organs are the flowers. In the flowers, there are stamens and the ovary. That is the style, stigma, and that is androecium and the gynoecium. Androecium, a male gametophyte, and gynoecium is a female gametophyte. In the plants, those germ cells are the stamens and the pistil. While in animals, it is sperm and the ova. So in plants, the reproductive organ is a flower. In that flower, stamens, in that the pollen grains, while and the ovary, which are the male and female gametes. While in animals, that is the sperm and the ovum. Here, before going that, I said that we'll deal with what is haploid and ploid. We have 46 number of chromosomes. Yes, in the somatic cells, in the body cells, it is a complete set of chromosomes. Those cells, somatic cells with a complete set of chromosomes are said to be as diploid, which is indicated by the symbol as 2n. Yes, the cells which have complete set of chromosomes are called as diploid cells. But the cells which have half the number of chromosomes, example, you take the sperm and the ova. These are the gametes. These are the reproductive organs or the gametes. They have only 23 chromosomes. 23 and 23 form an embryo which is having 46 number of chromosomes. So this condition with complete set of chromosomes is called as diploid, and the condition with half the set of chromosome is said to be as haploid. So in meiosis, the first process is as usual the prophase 1. We'll start with the prophase 1. Prophase 1 is divided into 5 substages. Leptotene, zygotene, pachytene, diplotene, and diakinesis. It is divided into 5 substages: leptotene, zygotene, pachytene, diplotene, and diakinesis. So, before the cell is entering the process of prophase, here it is the cell is under the process of interphase where there are chromatin network, nuclear membrane, the centrioles and everything. If you look into this, so the first we will start with the events that occur during leptotene stage. So what happens during the process of leptotene? So I will just repeat the processes. Those are leptotene, zygotene, pachytene, diplotene and diakinesis. Now, what are the main events that occur in leptotene stage? So, what is happening during leptotene? Condensation chromosomes. There is condensation of chromosomes. They coil in such a manner that they appear in the form of string of beads. The condensation is to such an extent that they coil. They just coil. I will just take the example. Chromosomes, they coil in such a manner that they appear as string of beads. The coiling is in such a manner that they appear as string of beads. Now they are known as chromomeres. They are known as chromomeres. So, chromosomes, they appear as, they get coiled and they get condensed in such a manner that they appear as string of beads. And now we call them as chromomeres. So in this chart you can see here in the leptotene all the chromatin network undergo the process of condensation. The chromatin material, yes, nuclear membrane is still there, nucleolus is also there, the centrioles are also there, but what happens is changes in the inside the nucleus, that is the chromosomes. High condensation of chromosomes occurs with the coiling, and those coiling is in such a manner that they appear as string of beads. Now we call them as chromomeres. So in the process of leptotene, there is chromosomes are visible. You can see them clearly. There is highly condensation or coiling and appear like bead string of beads. Yes. Next, after leptotene, it is zygotene. Second one is where you can go all the main events that occurs in the zygote. Before that, we will be familiar with some of the words in the process of zygote. Yes, that is homologous chromosomes. What is synapses and synaptimal complex. We will try to know the meaning of these words: homologous chromosomes, synapses and synaptimal complex. Now, generally we say that we are having 23 pairs. Why we call as 23 pairs? Because in our there are totally 46 number of chromosomes, and some they are not similar to each other. All we call it as a pair because they appear similar. So in the process you will see that there are some chromosomes which are exactly similar to each other. So such type of chromosomes which are similar to each other are known as homologous chromosomes. Those chromosomes which are similar to each other are called as homologous chromosomes. This is homologous to each other. This pair is these are homologous chromosomes. With the chromatics and the centromere. So those chromosomes which are similar to each other are known as homologous chromosomes, or generally this is a chromosome which are similar to each other. So such type of chromosomes are known as homologous chromosomes, and the pairing of homologous chromosomes is known as synapses. The pairing of the homologous chromosomes is known as synapses. So which are the main events that are occurring is synapses with the formation of synaptimal complex. Synapses and synaptimal complex. When they pair with each other, that pair is known as homologous chromosomes. So each of it have a centromere with 2 chromatids, and we call them as bivalents. We call such type of sets as bivalents. We call the pair, and the process where pairing of homochromosomes occurs is known as synapses. Then as they pair with each other, the chromosomes overlap on each other. They are just overlapping on each other. When they overlap on each other, they form a complex, and that is known as synaptimal complex. While in zygote, you can see that this is one. Here you can see in white string and a pink string. Yes. So these are the homologous chromosomes, and the pairing appears. There is pairing of homologous chromosomes. So the main events that occur during the zygote is the synapses and the synaptimal complex, where pairing of homologous chromosomes is known as synapses, and the overlapping of such pairs forms a complex and is known as synaptimal complex. Coming to the third stage. Patchy. Third is patchy. Now, in this process of patching, there is crossing over, then you will see at a point, there is a point where crossing occurs and is called as chiasmata, or singularly as chiasma. So this are the homologous chromosomes. Before going to the process, we will try to understand some of the terms. That is, what are sister chromatids, and non-sister chromatids. In this, these two are considered as, or these two are considered as sister chromatids, while these chromatids are known as non-sister chromatids. Yes, the chromatids of the same chromosomes are known as sister chromatids, and the chromatids of the two different chromosomes, are called as non-sister chromatids, where you can see that when synaptic complex is formed, there is overlapping of the chromosomes. Yes, with the help of a protein, yes, there is a protein complex, so that protein complex will help in overlapping of the chromosomes. Those chromosomes form a complex and is known as synaptic complex. When there is overlapping, there is a process called as crossing over. So, crossing over occurs between the non-sister chromatids. I will show the process in only one arm. That is the chromatids. Here, this process is known as crossing over, and there is a point, there is a part or a point where the crossing over occurs. That point is known as chiasmata, where the segments are being cut and again joined, or they are being segregated and again joined. They get separated, they are being broken and again joined with the help of the enzyme. So this enzyme helps in dissecting the segments. You can see the segments are being exchanged. There is exchange of the segments. As there is exchange of segments, you can also find that there is change in the genetic components, which is in the form of recombination. Why you are not exactly similar to your siblings is because of this process. When there is a process of crossing over, there is exchange of the genetic material. Their exchange of genetic material occurs by the process of crossing over, where there is breaking of a segment and also joining of the segment. So in patching you can see there is a process of crossing over, and the point where they are crossed is known as chiasmata, followed by diploid. Or you can see in this chart, there is these are the non-sister chromatids, where crossing over occurs between the non-sister chromatids. Yes, and the point where the segments are being broken, separated and again rejoined, that point is called as chiasmata. Then the next process is diploid. In the diploid, the process of crossing over occurs. So, in the process of diploiding, slowly the synaptic complex, that is the chromosomes that were overlapping on each other, it gets to dissolve. They start to separate, or we can say that the attraction of the chromosomes, homologous chromosomes, slowly dissolve and start to separate. When they start to separate, yes, slowly the chiasmata also try to deviate. It also starts to dissolve. So that process of slow dissolution of the chiasmata is known as terminalization. Where the two, after exchange of the segments, at the point of chiasmata, they start to separate from each other. So here the synaptic complex try to dissolve, and the chromosomal fibers, the chromosomes, they start to separate from each other, or we can say that the attraction between the chromosomes start to disappear. At the end, as there is crossing over, you will see that the point where it gets crossed over, you will find a new set of chromosomes. You can see the difference in the figure. Yes? Or if you find any references, you can just go through the figure or follow the NCERT textbook. So there is a process of chiasmata overlapping. The segments are being broken and again rejoined with the help of the enzyme recombinase. Yes? At the end of it, during the diploiding, slowly what happens? The synaptic complex starts to dissolve. The attraction also starts to dissolve. Slowly there is terminalization, where the chiasmata are being separated. They are being separated. Now you have a new set of chromosomes which have exchanged their segments, and these chromosomes are with the exchange of the segments. So as you can see in the process of diploiding, there is exchange of segments. Then a completely attraction loses everything is there. There is breaking of the segments, joining of the segments. Slowly terminalization occurs and the chromosome starts to repel from each other. Ultimately a new set of chromosomes. That marks the events of the diploidy. In the last, it is diakinesis. The process is known as diakinesis. In this process of diakinesis, complete terminalization of chiasmata occurs. Completely. Slowly the chiasmata starts to dissolve. You can see there is complete terminalization of the chromosomes where you will not find the chiasmata. Complete terminalization of chiasmata occurs with the formation of new sets of chromosomes. Yes, slowly what happens? The nuclear membrane, remember the steps of the prophase. The end of the prophase steps starts to develop. The nuclear membrane, nucleolus, the centrioles produce the aster fibers. This asters starts moving towards the poles. Then nuclear membrane, nucleolus starts to disappear and the chromosomes are free in the cytoplasm. So there are 5 sub-stages. After entering the interphase, it enters into leptotin. Main events is, in this, the chromosomes are appearing in beaded like forms, as a string of beads, and are called as centromeres. While in zygote, the process there are homologous chromosomes. Those that get paired and are known, that process is known as synapsis, and synaptic complex is being formed. While in PARVATI, crossing over occurs. There is exchange of the segments. With the exchange of segments, that will lead to recombination. There is exchange of genetic material. Then in the process of diploidy, slowly there is repulsion among the chromosomes. They start repelling, or the attraction dissolves and starts moving, and even the chiasmata also dissolves, where you can see new set of chromosomes are being formed. Then in the process diakinesis, yes, there is complete dissolution. The terminalization occurs, is completed with the formation of new set of chromosomes, and all the chromosomes are free in the cytoplasm. Then this is about the prophase, where nuclear membrane, nucleolus and other all organelles disappear, and the centrioles. The centrioles produce the aster fibers. Then it starts with first metaphase. As in the first metaphase, it is all the chromosomes lie at the center. The centrioles are towards the poles. They are producing the spindle fibers. The spindle fibers get attached at the centromere of the chromosomes, and all the chromosomes are at the center. So it is regarded as metaphase, followed by anaphase. In the anaphase, 2 daughter chromosomes are being formed. They are being pulled by the spindle fibers contract, and the 2 sets of chromosomes are towards the poles. Again in telophase, with the help of cleavage furrow, a new cell membrane is being developed between the 2 daughter nuclei, and the process are similar to that of telophase. The nuclear membrane, nucleolus, appears simultaneously with, after chirokinesis, it begins with cytokinesis, where 2 daughter cells are being formed. 2 daughter cells are being formed. Yes, so this is the first meiotic division. And in the second meiotic division, the same process in the second meiotic division, that is karyokinesis 2. Again prophase 2, metaphase 2, anaphase 2 and telophase 2. So with the chart we will go with to understand the second process of meiotic division. So in the first meiotic division, after the end of meiosis 1, the cell, two daughter cells are being formed and they are at the rest. Before starting with the process, they are at the rest. That process is known as interkinesis. Again it starts with the same process, that is prophase, metaphase, anaphase, telophase and telophase. So here the prophase, it is in mitosis it occurs only in the one cells, but here both the daughter cells enter the process, that is the prophase 1. Again in prophase you will see that there is dissolution of nuclear membrane, the nuclear membrane, nucleolus and other cell organelles starts to disappear. The centrioles in each of the cell start moving towards the poles, and at the end of prophase you will find two sets of centrioles with the fibers, spindle fibers at the poles, astral fibers at the poles. Then the chromosomes are freely suspended in the cytoplasm. Then followed by metaphase. The centrioles produce the two fibers, continuous fibers, those which extend from one centrioles to the other centriole, while the chromosomal fibers, those fibers which get attached to the centromere. In both the daughter cells you will see that all the chromosomes are lying at the center of the cell, and it is marked by the presence of chromosomes at the center of the cells, that is metaphase. Then in anaphase you will see there is splitting of the chromosomes. There is splitting as they split. Yes, two daughter cells are moving towards the poles. In anaphase, splitting occurs where the daughter cells, that is the dyads, move towards the opposite poles, followed by telophase. In telophase you will see that again on each set of chromosomes nuclear membrane, nucleolus starts to develop with the formation of cleavage furrow, and at the end four daughter cells are being formed. First two cells and it divides to form two cells. Totally four daughter cells are formed which are haploid, that is N with half the number of chromosomes. Yes, for cleavage cytokinesis occurs with the changes in the nucleus, followed by karyokinesis occurs, then followed by the process of cytokinesis, with then four daughter cells are being formed, and each of these daughter cells or the divided cells, the cells are converted, they undergo the process of spermatogenesis where they convert themselves into sperm or ovum in the reproductive organs of the organism. Yes, so at the end of meiosis you will find that the four daughter cells, same process but different difference is that these daughter cells that are being formed by the process of meiosis are haploid. Haploid number of chromosomes, that is half the set of chromosomes. For example, if you take, it is each of them have only 23 number of chromosomes, that is the sperm have 23, ovum have 23. Then in the process of reproduction they develop to form a zygote which have 46 number of chromosomes. So in meiosis there are two divisions, meiosis 1 and meiosis 2, followed by karyokinesis. In that the prophase is of longer duration or the longest duration, where we have studied the 5 sub-stages. So at the end of the process of meiosis, 4 daughter haploid cells are being formed, and this process is called as reductional division. So only it is called as reductional division, where 4 haploid cells which undergo the process of spermiogenesis, where they convert into the reproductive organs as the sperm and the ovum. In the tomorrow's class, we will go with significance of meiosis and what are the differences between the mitosis and the meiosis. Thank you.